Hello, everyone, and welcome to our online learning segment for this week. If you recall, last time in class, we had a chance to look at the brain, and specifically, you got to do some partner exploration related to the brain. And we spent some time looking at some major brain structures as well as their functions. How do those particular parts of the brain influence our thoughts, emotions, and behaviors? Last time, we spent most of our time together looking at the hindbrain and the midbrain. And we talked about how the hindbrain controls for really basic bodily functions that we share in common with other animals. We talked about how the midbrain controls a lot of sensory integration. And we spent a little bit of time looking at specific structures within the forebrain, that part of the brain that makes us uniquely humans and allows us to do complex critical thinking and things of that nature. Today, we're going to spend some specific time looking at the cerebral cortex in general. And we're also going to look at how the brain exhibits contralateral control over the body and some other really fascinating things about the brain that we've learned through case studies. If you remember in our research methods chapter, a little throwback to that chapter, we talked about how case studies can be particularly useful when you want to study something about someone that would be completely unethical to do to that person. And so we do know a lot about the brain from various case studies. So all of that said, let us dive into the fun together here. For this particular portion of information, you're going to be completing your interactive notebook number 4. So you want to make sure that you have that open and ready for yourself. And again, for the first little bit here, we're going to do an overview of the cerebral cortex where you can simply add in some information that's articulated for you in your interactive notebook. All right, so the largest and most developed area of the brain, your cerebrum, refers to most of your brain except for the hindbrain, the brainstem and cerebellum, and it's comprised of 2 separate layers. So the internal layer of your cerebrum is made up of axons of neurons and glial cells and is called white matter. I like to think of it, I know this is weird, but I like to think of it as a cupcake. So if you can see my hands on the screen here, here's your cerebrum, okay? Then on top of the cerebrum, there's a 1/4 inch thick wrinkled outer layer that's called the cerebral cortex. I like to think of it as the frosting on top of the cerebrum, which is the cupcake. And your cerebral cortex is made up of the cell bodies of neurons called gray matter. The cerebral cortex then is responsible for sophisticated thinking and learning in humans. And any portion of the cerebral cortex that's not devoted to particular motor or sensory functions is known as an association area. And an association area is basically just an area of your brain that helps communicate with other parts of your brain. So the rather large cerebral cortex in humans, which is about the size of a newspaper if it's stretched out flat, it's folded up in itself to fit in your skull, obviously. And the ridges along the surface of the cortex are referred to as gyri, and the valleys on the surface are called sulci. The 2 halves of the brain are referred to as the left and right hemisphere. Especially deep or pronounced grooves on the cerebral cortex in each hemisphere are called fissures, and several deep fissures divide the cerebral cortex into 4 regions or lobes. We have the frontal lobe, which is right behind your forehead. So if you tap your forehead, you're basically tapping the front of your frontal lobe. Your skull's separating it, obviously. The parietal lobe is located directly behind the frontal lobe. The temporal lobe is located right above the ears, and the occipital lobes are right at the very back of the cortex. As you work through your interactive notebook today, we're going to go ahead and pause the video. And what you're going to spend a little bit of time doing is exploring these different lobes of your cerebral cortex, so we can figure out what are some specific structures and their functions in those regions that help control our thoughts, emotions, and behaviors. And rather than me just talk with you about those things, want to give you a chance to actually dig in and explore those using a really cool interactive website. So if you would, let's take a look at your interactive notebook itself. On this particular page, you're going to click on the link there, and it's going to take you to this really cool interactive website where you can explore all of these different structures of the brain and their functions. So you're going to go to this website while having your notebook open. And for each of the areas or structures that you see here, you're going to go ahead and just explore what is the function of that particular area or structure using that interactive website, and you can write down what you find. So for instance, if you go up to the left-hand side here of this website and click on the down arrow, you'll be able to find any of the structures that are listed in that particular advanced organizer in your chart. You can go ahead and click on something like Broca's area, let's say, and it's going to rotate the model so you can actually see that area of the brain, and it'll tell you all about the function of that particular area of the brain, as well as give you some fun, interesting facts. So we're going to pause the video for quite a bit of time so that you have a chance to explore on your own these particular areas of the frontal lobe. You're going to focus on the prefrontal cortex, Broca's area, and the motor cortex. The parietal lobe, you're going to focus on the somatosensory cortex. The occipital lobe, you're going to focus on the visual cortex. And then the temporal lobes, you're going to focus on the auditory cortex and specifically Wernicke's area. So go ahead and take a moment to do that, and then we will continue on with our time together. All right, so now that you have a sense of how the different lobes of the brain have particular structures that influence function, let's go ahead and talk about contralateral processing. So this is fascinating stuff. So when we think about the frontal lobes, the hemispheres of the brain control the contralateral or opposite sides of the body. So that just means that the left side of your brain actually controls the right side of your body, and the right side of your brain controls the left side of your body. The left cerebral hemisphere is responsible for processing sensory information from the right side of the body and vice versa. So tactile sensation from the left side of your body is actually processed by the right side of your brain at the somatosensory cortex. And that tactile information is transferred in the spinal cord or the brainstem. When we think about vision, objects on the left side of your visual field in both eyes are processed on the right side of the visual cortex, and visual information is transferred at the optic nerve. It's a big fancy word for nerve that you see there. The left cerebral hemisphere is also responsible for processing motor information for the right side of the body and vice versa. And muscular contractions then are coordinated by the motor cortex. So just lots of different examples of how the brain exhibits contralateral control to influence our thoughts, emotions, and actions or behaviors. Okay, all people with intact brains use both hemispheres and have access to the abilities that are dominantly controlled by each. Again, because the corpus callosum connects those hemispheres together so that all the different sides of both different sides of your body can work together with your brain. But there are some cases where we've had to split the corpus callosum for a particular reason, typically to treat severe epilepsy. And we refer to those case study patients as split brain patients. And they've taught us a lot about brain lateralization. And the researchers that you see here, Sperry and Gazzaniga, are famous for this particular research. Okay, so brain lateralization or split brain patients. Let's take a look at this. Split brain patients do not display differences in intelligence or personality, but the lateralized functions of each hemisphere, the left and right hemisphere in the brain, can be observed in specific lab experiments. So for instance, and you can see an image of this, what I'm describing to you on the right-hand side of the slide here. If a word is flashed to the left visual field, that's received by the right side of each eye, this information is sent by the optic nerve to the right hemisphere. Normally, this information would then cross the corpus callosum to the left hemisphere where language exists. But because this connection has been cut, the split brain patient is not able to say the word for what he sees out loud, or she. However, the nerves of the right motor cortex cross under the medulla and will allow the split brain individual to use his or her left hand to actually select the object that was viewed. So fascinating. If a word is flashed to the right visual field, which is received by the left side of each eye, then information will be sent to the left hemisphere, and the split brain patient will be able to pronounce the word because the left hemisphere is in charge of language. Bonus points if you can remember the name for the speech area that's located in your left hemisphere. Okay, so if we looked at the left hemisphere here, sensory stimulus, it receives sensory stimulus from the right side of the body. It exhibits motor control of the right side of the body. It's in charge of speech, language, and comprehension. It plays a dominant role in analysis and calculations, time and sequencing, and recognition of words, letters, and numbers. Whereas your right hemisphere, again, controls sensory stimulus and motor control of the left side of the body. And it's more active in things involving creativity, spatial ability, context and perception, and recognizes of faces, places, and objects. Okay, just to give you a more real world view of this particular phenomenon of split brain patients, go ahead and pause the video, this particular video, and check out the watch this video that we've linked here related to split brain patients. I think you're going to find it really fascinating. So go ahead and pause and do that. All right, paying a little bit more attention to our language association area. So we have Broca's area and we have Wernicke's area, okay? Let's do a little bit of a deep dive here. So in 1861, Paul Broca performed an autopsy on the brain of a patient nicknamed Tan, who had lost the capacity to speak, although his mouth and vocal cords weren't damaged, and he could still understand language. And so they were trying to figure out, why has he lost the capacity to speak? So Tan's brain in particular, it showed deterioration of part of the frontal lobe of the left cerebral hemisphere, which is now named Broca's area, as did the brains of several other similar case study patients. And so we learned over time by doing those case studies, linking brain structure to a loss in function, that Broca's area in the left frontal lobe controls our ability to speak. Okay. We also have a Wernicke's area located in a different part of the brain, and you can see that on the diagram there. So in 1874, Wernicke was looking at related nerve diseases to specific areas of the brain, and he discovered that the structure in the left temporal lobe controls language comprehension, named it after himself, and called it Wernicke's area. So this caused a loss of the ability to comprehend written and spoken language when damaged, where patients would try to speak, but they would speak in garbled sentences and they couldn't understand what other people were saying. So to kind of wrap this up together here, Broca's area, it's all about producing language. If you have damage to Broca's area, you're unable to produce your own language. If you have damage to Wernicke's area in your temporal lobe, you're not able to understand the language that other people are saying to you. Two super important areas that help us communicate, obviously, in the world around us. Okay, so that's a little bit about the cerebrum, the cerebral cortex, all of those parts of the brain that help make us uniquely human, and some specific structures and functions within. Now, we're going to turn our attention to some tools for examining brain structure and function, because obviously it's hard to get in there and see what's actually happening. So we have some windows on the brain to talk about today, or some tools that we can use to really see what's going on in there. Let's briefly talk about early methods of brain research, and then we'll kind of move forward with the technology tools that we have today to look at different parts of the brain. So early brain research typically involved looking at case studies of individuals who suffered brain damage, and then looking at how they acted in the world to see if there was a connection between what part of the brain was damaged and what we noticed in terms of somebody's thoughts, emotions, and behavior. But thanks to advances in biological psychology, we have a lot of new ways of looking at the brain. But here's some old ones. So the first one is accidents, and doing case studies of people who have undergone serious accidents or unfortunate events. And so we talked briefly about Phineas Gage in that first chapter that we looked at together. This unfortunate man, you can see a picture of him right below my face in the top right of the video there he actually is in real life posing with the metal rod that blasted through his skull, and yet he continued to live. And through his case study, we learned that the frontal lobes regulate emotion and the ability to predict and play into future events. Tan, the case study of Tan that we just looked at with Broca's aphasia, is another example of a case study. Another way that we study the brain is looking at lesions. And so a lesion is basically the removal or the destroyal of a small portion of an organism's brain. This is unethical to do to human beings, obviously. It is considered to be ethical at times to perform these procedures on animals because the people doing these experiments claim that the benefit to human life outweigh the pain, suffering, and cost to animal life. So interesting thing for you to think about. What do you think about ethics related to that? Anyway, when we do lesion studies, we destroy a part of the brain so that we can see what is the resulting impact on human thought, emotion, and behavior. Direct stimulation is similar, but it's not destroying part of the brain. It's literally just stimulating a part of the brain to see what happens in the person's thoughts, emotions, and behavior as a result. So, for instance, stimulating the amygdala of mice results in an aggressive behavioral reaction. Something that I saw that was fascinating one time, I watched a video of a person undergoing surgery for a brain tumor, and the brain tumor was really close to where they thought that person's Broca's area was in the brain, and they wanted to make sure that when they removed the tumor, they weren't also going to destroy Broca's area, or else that person wouldn't be able to speak moving forward. And so they actually did an awake brain surgery on this 12-year-old patient who was so brave. They gave them all this anesthesia, and they allowed the mom to be in the room with the patient, holding the patient's hand. But literally, the patient was awake while they were having brain surgery, and they stimulated different parts of the patient's brain to find Broca's area. So for instance, they stimulated one part of the brain and they said, say the alphabet. And so the person was like A, B, C, D. And then when they stimulated the actual Broca's area in the brain, the person wasn't able to form words anymore. It was like A, B, C, D, H, I, J, And so they found Broca's area in the brain and tagged it with a little sticker. And then they made sure not to remove that particular spot in the brain. So amazing what we can do with brain research today. And then, of course, split brain research. We talked about that. We also can learn about the brain through autopsies and brain imaging. So let's spend a little bit of time looking at different brain imaging techniques. And for these techniques, the AP exam, they love to ask you whether the brain imaging techniques help you examine the function of the brain, what the brain's actually doing, the structure of the brain, like a still life picture, or both. So let's take a look. One brain imaging technique is called an EEG, or electroencephalogram, and this measures electrical activity of neurons below a series of electrodes that are placed on the scalp. And EEGs are often used to show brainwave patterns of electrical activity during different stages of sleep, and especially during seizures, so they can see what parts of the brain are active and how they might treat those. So the purpose of an EEG is actually to look at brain function. Advantages of an EEG, it's non-invasive, and it does help us see abnormal patterns that would indicate neurological disorders that need to be treated. Disadvantage of an EEG is that it's really difficult to determine which specific brain area is producing the electrical activity that you see on the output from an EEG. Okay, a PET scan is another brain imaging technique that allows us to look at the function of the brain, but a little bit differently than an EEG. So on this previous slide here, this rectangle with brain waves that are outputted on there, that's what an EEG reading looks like. And so it does tell us about energy and function of the brain, but it's not very specialized. Contrast that with a PET scan. Okay, so a positron emission tomography scan, a PET scan, involves injecting a small harmless amount of radioactive material like glucose into the bloodstream. And then the PET scan, the sugar, the glucose in that injection, it travels to your brain and it lights up different areas of the brain that are particularly active during particular tasks. So, for instance, you can volunteer to do a PET scan, and somebody might say, okay, now start writing an essay. And as you're actually doing that function in the world, the part of your brain that's metabolizing glucose shines really bright. So you can see, again, under my face here in the corner, those areas of that PET scan that are dark red and bright orange are the most active areas of the brain. And this really allows us to see the link between brain activity in real time and functions that people are doing in the world. Advantages is that this allows us to investigate different mental illnesses and neurological problems by illuminating that link between the part of the brain and the function in the world. Disadvantages, it does expose patients to low levels of radioactive material, and it is difficult to pinpoint the exact location of brain activity, but you can see how much more useful this would be for looking at function than even an EEG, let's say, if we're looking at specific disorders and things like that. We also have a CT scan or a CAT scan. This is a computerized axial tomography, and it creates specific X-rays of the brain. When the individual is in the machine, the X-ray tube rotates around the body, taking a series of X-rays. And so the CT scan is often used to locate tumors or other types of abnormalities that are affecting brain structure. So the CAT scan looks at structure as opposed to function. It doesn't tell us what parts of the brain are active at any given time. It just takes a really sophisticated X-ray that lets us look at the structure of the brain in general. Advantages is that it can expose large brain abnormalities, and it's much more sensitive than just a traditional X-ray image. Disadvantages, it involves radiation, and it's not capable of locating really tiny brain abnormalities. Typically, by the time something shows up on a CAT scan, it's relatively advanced or progressed. And we also have an MRI, and again, this looks at structure as well. It's called magnetic resonance imaging is what it stands for, and it uses strong magnetic fields that cause different molecules to vibrate at different frequencies, which produces detailed images of slices of brain tissue. Advantages here: MRIs generate images of brain structures with greater clarity than the CT scan, and there's no exposure to radiation or radioactive materials. Disadvantages, though, can't be used on an individual with any sort of metallic implant, including a pacemaker or even a surgical pin. And the individual is required to remain really still for at least 45 to 60 minutes. They cannot move at all. And so some people mentally just genuinely can't handle the process of going through an MRI. You're in a tiny little tube like you see there, and you just can't move at all. Lastly, then we have the fMRI, and the fMRI looks at both structure and function. It's called a functional magnetic resonance imaging technique. And just like the MRI, it uses magnetic fields to produce images of the brain, but it also tracks real-time brain activity by measuring the blood flow carrying oxygen to active brain tissue. Advantages to this method are that there's no exposure to radioactive materials like during a PET scan. And so you can do multiple scans on the same individual because there's no worry of that. It also allows us to pinpoint and track mental processes that occur over seconds as opposed to just minutes. Like thinking about an object, you literally can see different parts of the brain light up as the person is engaging in thought. So cool. Disadvantages, it can't be used on individuals with metallic implants. And although there's no harmful consequences that have been identified so far, the long-term impact of exposure to these powerful magnets is really not well researched. And so it's a little bit unknown. So those are some different windows into the brain. Want to share with you one last really interesting concept called neuroplasticity, and we'll unpack this a little bit more in class next time together. But one of the most amazing abilities of the human brain is the capacity that it has for adaptation or plasticity, regrowth. which allows it to modify itself if it's been damaged. For example, if a particular area in the brain is damaged, nearby areas can learn to assume the same functions of the regions that were destroyed by developing new connections between dendrites. So, neuroplasticity is literally new connections that are being made between neurons in your brain, um, typically to adapt to function that's going on in the world. So it's your brain's ability to reorganize itself by forming new neural connections throughout life. Neuroplasticity allows neurons in the brain to compensate for injury and disease and to adjust their activities in response to new situations or to changes in their environment. And for those of you who are further interested in this, I put two particular videos here that are beyond fascinating. I highly recommend you watch them, particularly the story of Jody, and we'll talk about her a little bit together in class next time. And then finally, to end our time together for this particular learning, I put an article here for you that says, what is brain plasticity and why is it so important? So what I would love for you to do for your final online learning activity associated with this section is go ahead and pause the video, or just end the video actually. You can go ahead and read that article and answer the questions that are listed here. This is truly just for your own learning and knowledge. You don't need to worry about writing incomplete sentences or anything like that, but just get the main ideas down in each box. And this will help you as you continue to think about brain plasticity and apply it to real life. Thank you so much for your time and effort in this particular area, and I look forward to seeing you in class next time.